So hello viewers, welcome to my channel Ronnie Nerd Sciences, and here today I'm with my new video. So don't forget to like, share and subscribe for more interesting videos. In this video we are going to see the phenomena of haematopoesis. Haematopoesis, as the name suggests, hemato refers to blood and poiesis is formation. It refers to the commitment and differentiation process of stem cells to different type of cells we found in the blood. For example, red blood cells. Now hematopoiesis, which is the making of these different cells, they occur mainly in the bone marrow, and this is particularly in adults. This referred to as medullary hematopoiesis. But hematopoiesis can also occur in tissues such as the liver, thymus and spleen, and this is called extramedullary hematopoiesis. And we learn about that in this video. So let's begin by zooming in the bone marrow. Now we are beginning by zooming in the bone marrow, and in the bone marrow we can find the hematopoietic stem cells. These stem cells can first of all differentiate into either what is called a common lymphoid progenitor cell or common myeloid progenitor cell. The common lymphoid progenitor cells then further differentiates into different lymphoblasts with different genetic profiles, and this will allow them to form and mature into different type of lymphocytes. So here I am just drawing the blood stream just as the cells exit the bone marrow and will enter the blood circulation. So these different lymphoblasts then with different genetic profiles will become either naive B cells, pre-T cells or natural killer cells. Now also common lymphoid progenitor cells can differentiate into certain kinds of dendritic cells. The dendritic cells, when it is formed, will move into tissues and become known as lymphoid dendritic cells. Dendritic cells are peripheral antigen presenting cells that are actually very important in connection between innate immune system and adaptive immune system. So that was the lymphoid progenitor cells lineage. Now let's look at common myeloid progenitor cells. And it is important to know that outcome of these lineage cells that can circulate in blood. So hence hematopoietic, again from the hematopoietic stem cell, it can become a common myeloid progenitor cell. And there are different maps of hematopoiesis that depict common mild progenitor cell differentiation. But now I'm going to show you what I think is the easiest. Anyway, the common myeloid progenitor cells can become myeloblast and eventually become granulocytes. These granulocytes are the cell containing granules, and they can either band neutrophils, band basophils and band eosinophils. And these granulocytes are band cells because they are immature cells. But once they enters the blood circulation, they become mature and what we know as neutrophils, basophils and eosinophils respectively. Neutrophils, basophils and eosinophils. The neutrophils are important in acute response to infection and inflammation. Basophils are important in allergy and parasitic infection, and eosinophils are also important in allergy and sensitivity. The common myeloid progenitor cell can differentiate and become pro-monocyte and mast cell precursors. The pro-monocytes then mature and become what we know as monocytes. They are essentially circulating macrophages, and so once they move into a tissue such as the skin, they become tissue macrophages, and macrophages are antigen presenting cells, which again are important in the connection between innate and adaptive immunity. The monocytes or pro-monocytes can also become dendritic cells, and if they become dendritic cells in tissues, they refer to as the myeloid dendritic cells. So again, we have two important antigen presenting cells, macrophage cell and dendritic cells. The mast precursor cells become mature mast cells once they enter circulation and move into tissues. Mast cells are very important cells in allergy and hypersensitivity. Finally, the common myeloid progenitor cells. Now we are going to discuss about the myeloid progenitor cells. These cells differentiate and become megakaryocytes with stimulation of thrombopoietin, a hormone produced by liver and the kidneys, or the myeloid progenitor cells can become erythroblast through stimulation of erythropoietin, a hormone released by kidneys. Let's look at megakaryocytes first. They are normally present in bone marrow, not in circulation, but these guys give rise to platelets we found in blood, make a carrier site of rupture, releasing place and platelet in circulation, and platelets are very important in clotting mechanism. Erythroblasts are still nucleated red blood cells, meaning that they have a nucleus, but once the erythroblast enter circulation and mature, they become erythrocytes, which are a nucleated cell. Erythrocytes are the red blood cells. Hope you got the process. Thanks for watching the video.